Hey y'all, welcome back to another genetics video. Today what we're going to go over is DNA replication for chapter 6. So today we're going to go over the various enzymes used to replicate your DNA, the differences between DNA polymerase 3 and DNA polymerase 1, and we're going to go over a critical thinking question that you typically may see on an exam, which is basically, why do we have a leading and a lagging strand? And that'll be basically for today. So to begin with, we're going to talk about the enzymes used for this process. I have basically the step-by-step tutorial that the book provides for students, but essentially what happens here is that you have something called a helicase. And helicase is basically a protein that unwinds the double-stranded DNA into single-stranded DNA, and this protein essentially allows for the replication bubble to be extended. You have something called a topoisomerase. So this protein is basically used for, especially in circular DNA, because circular DNA typically during replication, it supercoils into a really tight coil, and that can be problematic for DNA during replication because that supercoil can eventually snap the DNA in half. So the topoisomerase is used to prevent that from happening, and basically what topoisomerase does is it finds that supercoil, it will nick the DNA at that supercoil, it will unwind the DNA back to a linear form, and then it will reform that phosphodiester bond, and then and that's how the topoisomerase works. You have another enzyme over here not listed here, but to actually develop the replication bubble initially, you need a protein called DNA-A. So DNA-A finds OraC, and then when DNA-A finds OraC, it will break apart the double-stranded DNA into single-stranded DNA. Okay, so that's basically for step 1. Step 2 is you're going to use something called a single-stranded binding protein, or SSBPs. So basically all these do is they bind to the single-stranded part of the DNA, and they prevent the double-stranded DNA from reforming again. So that's all it does. Then you have something called a DNA primase. What this does is that this primase enzyme adds RNA primers to the DNA, and then these primers are utilized by DNA polymerase 3 to synthesize the new strand. Without the RNA primers, DNA polymerase 3 does not function properly, because you need a functional hydroxyl group on the 3' end of every DNA nucleotide in order for you to replicate. So if you never had an RNA primer in the first case, you would never be able to make a functional or a working phosphodiester bond. So therefore you need the RNA primer to be there. Okay? The fourth step, which I already mentioned, is DNA polymerase 3 continues the replication going forward in 2 different ways, in a leading strand and a lagging strand, which we're going to talk about a little bit later. So during this process, the leading strand is basically going to be synthesized continuously, and it won't stop. But then the lagging strand, however, because it's basically going in an inverse direction, it has to be replicated in segments, right? As you can see here. So what's happening here is that at every particular point, you're adding a new RNA primer here to allow for replication to occur. During this time, whenever you're adding a new primer in the RNA polymerase 3 displaces onto the lagging strand again, what's happening here is a DNA polymerase 1 is being added to the lagging strand, and the DNA polymerase 1 is basically going to remove all that junk RNA, and it's going to add it's going to add new DNA in its place. And then lastly, what's going to happen here, DNA ligase is going to come in and it's going to reform the final phosphodiester bond between the last two DNA nucleotides, because the DNA polymerase 1 doesn't actually reform the final phosphodiester bond. Okay? So basically that was a quick rundown of DNA replication. I guarantee you've probably already heard it like 5 million times, you know, in Biology 1 and like other courses like that. So we don't need to spend that much time in it for our purposes. But for our purposes, what we need to figure out is the differences between DNA polymerase 3 and DNA polymerase 1. Both of these DNA polymerases have a proofreading function, but one of these DNA polymerases has an additional proofreading function that allows it to function. So basically, the main difference between these is that DNA polymerase 1 has the ability to do something called a 5' to 3' exonuclease activity. And I might as well write it down. And then it also has an ability to do a 3' to 5' exonuclease activity. While DNA polymerase 3 only has the function to do a 3' to 5' exonuclease activity. So what are these nuclease activities used for? These nuclease activities are used to repair a DNA replication error, because most of the time, statistically speaking, these enzymes are going to make a mistake sometimes, and they're going to add an incorrect nucleotide where it's not supposed to be found. So you need these exonuclease activities to remove the nucleotide that you've added and add a correct functional one that's supposed to be there, which dramatically reduces the chances of you developing a mutation at that point. So what is this 3' to 5' exonuclease activity? So this 3' to 5' exonuclease activity is essentially, let's say in the red one, this red strand is basically the parental strand, and the white strand is a newly synthesized strand. So this would go 5' to 3', and then the bottom strand is obviously 3' to 5'. So let's look at it as perspective from DNA polymerase 3. DNA polymerase 3 would be right over here, and it will continuously add new nucleotides going from 5 to 3. Sometimes it may make a mistake, and it would accidentally add a A where you should have added a C here. Okay? So in order for you to correct that mistake, what you would need to do is you would need to use your 3' to 5' exonuclease activity. So you would remove the nucleotide. From 3 to 5', and that's why it's called a 3' to 5' exonuclease activity, because essentially the DNA polymerase is basically in front of the hydroxyl group on the 3' end, and then if it senses that there is an incorrect base pair attached in the newly synthesized strand, it's going to sense that and it's going to remove it all the way through the 5' end. Okay? So basically that's all 3' to 5' is. And then you can use the exact same idea for the 5' to 3' exonuclease activity. Oops. Let's say that was a C. So let's say over here we have our RNA primers, and we're going to use this example for the lagging strand. If we have our RNA primers here, this end over here, what we're staring at is essentially the phosphate group on the 5' end. That's basically what the DNA polymerase 3 is going to, or the DNA polymerase 1 is going to be looking at on the lagging strand. It's going to sense that you have an incorrect base pair on the 5' phosphodiester bond, or it's going to sense that there's RNA. So basically, this DNA polymerase 1 can remove that section of RNA from going from 5 to 3', and that'll be the differences between those two. So make sure you use these. It can get a little bit confusing on remembering which one is which, but just remember DNA polymerase 1 has both of them. DNA polymerase 3 has only the 3' to 5'. And if you get confused, imagine you're the DNA polymerase, and you would use these notations to figure out what you're actually removing during the proofreading function. Okay, that is it for that. Next up, what a lot of people struggle on is figuring out the question of why do we actually have a leading strand and why do we actually have a lagging strand? Now, a lot of people struggle on this problem because they pretty much state all the things that you need to state, but they don't necessarily write the way how you're supposed to be writing it. So they state that the lagging strand is synthesized discontinuously, whereas the leading strand is being synthesized continuously, which is correct, right? But they're not including the information that you need to include in order for it to be understandable for an exam. So the way how you should answer this question is, first of all, you want to talk about the DNA polymerase activity. So to begin with, DNA polymerase 3 has the way how DNA polymerase 3 works, or any DNA polymerase. It works by reading the parental strand it reads the parental strand from 3 to 5', and it builds new DNA from 5 to 3. So that's the first thing that you should include. You should always include that these DNA polymerases, they work by reading the polarity of the DNA, right? But the issue here is that because the polarity of DNA is anti-parallel, which means anti-parallel just means that one of your strands has a opposite directionality compared to the complementary strand. Therefore, these DNA polymerases will be working in opposite directions, right? Because they work in opposite directions, now you have something called a leading and a lagging strand, okay? And because of these leading and lagging strands, because they're working in opposite directionality, one of these strands is going to be leading, which is going to be continuous, because it fully synthesizes and it follows the directionality of helicase. But then you have a leading or a lagging strand that essentially is going backwards away from the replication fork, going backwards, and then because it's going backwards, you need multiple RNA primers, and you need the abilities of DNA polymerase 1 and DNA ligase for this to occur. So just remember the reasoning why you need a why do you have a leading and a lagging strand. But other than that, DNA replication in genetics is pretty much self-explanatory. If you put in a lot of effort to remember all the different terms, especially if you're not familiar with it, you should be okay. But just work around trying to answer some different critical thinking questions. But the most important one is, why do we have a leading and a lagging strand? But that's basically it. Hopefully this video helped you out. And yeah, that's it.